Good afternoon to all. I am Shweta from PG and Research Department of Microbiology. Now I am going to discuss about pure culture techniques of microorganisms. So first culture. What is culture? The microorganisms that are cultivated is known as culture. That is classified into two types, mixed culture and pure culture. What is mixed culture? More than one microorganisms, that is called mixed culture. Pure culture containing single species, that is called pure culture. Assessment of purity of culture. Several isolated colonies of a pure culture show the same cultural characteristics on a particular media. When stained and observed under the microscope, they look alike, the same stain and morphology. Several isolated colonies perform identically as in the case of similar biochemical results. Cultural and morphological characteristics. See here two pictures shown on slide. First one is mixed culture, that is a gram positive and gram negative microorganism mixed and shown under the microscope. Here another one picture, pure culture, single colonies, shown only single colonies. Biochemical characteristics, first picture shown mixed culture, second picture shown pure culture. Importance of pure culture. A pure culture can be grown, identified, characterized and tested. The physiology and the clinical aspects can be studied. Same results are obtained for a particular culture irrespective of number of times the test has been done. The rate of spontaneous mutation is low and the colon of a pure colony is identical in all aspects. History of pure culture techniques. Robert Koch was the first to perfect the technique of isolating bacteria in pure culture. He also isolated Bacillus anthracis that cause anthrax. Agar was first used in microbiology in 1882 by a German microbiologist Walter Hess working in Koch laboratory. Koch assistant Julius Richard Petri in 1887 developed the Petri plate, a container used for solid culture media. This picture shown a Petri dish and Petri plate with media. Techniques involved sterilization, dispersing the individual cells, thinning the sample. Sterilization techniques, moist heat sterilization, dry heat, filtration, flame sterilization, and biosafety cabinet. So first one autoclave, second one water oven. Autoclave used for sterilization, water oven used for glassware, dry heat sterilization, sets filter, flame and laminar airflow. How to isolate of pure culture or separate pure culture? By streak plate technique, pour plate technique, serial dilution, spread plate technique and enrichment method. So this is a streak plate techniques. The plate contain a mixed culture, then prepared media test tubes or plates. We have to take on separate one colonies from the plates and streak on the fresh test tubes, media containing test tube, and allowed for incubation. So this is a pure culture. From the mixed colonies, we have to separate one culture by streaking. The picture shown a pure isolated colony. Various streaking method for a pure culture separation, quadrant streak, continuous streak, radiant streak, etc. used for pure cultures. So this is a pour plate technique. We have to take bacterial sample mixed with warm agar, sample poured on sterile plate, and then allowed for solidification. After solidification, plate allowed for incubation. After incubation, colonies were observed in the plate. This is the serial dilution. After serial dilution, plates was incubated. After incubation, colonies were observed in the Petri plates. Spread plate techniques. We have to use the LRA to spread culture clockwise and anti-clockwise, and the medium. After it allowed for incubation, that plates after incubation, the plates containing colonies are absorbed. Enrichment method. Basal medium is incorporated with blood, antibiotics, growth supplement to enhance the growth of the desired culture. Maintenance of pure culture. Once the pure culture is isolated, maintenance and preservation is necessary for further study and application of the culture. Thank you.